Yeah, Sabrina. Hi, nice to meet you. Looking forward to meeting you. So then you know like the back of your hand.
Yeah.
Yeah.
So right over the forward.
Okay.
And that was red buttons away.
For the presentation.
Ooh, I think fun. We're fun. And then probably just. They have already. Thank you so much. Did you find this thing? Yeah. You find it. You didn't find one?
He grind it.
You're out there, right?
No, I pressed it on. That's like. No, I don't know. You have to do that. I don't know what I'm because here does the you can make I think
Monitor. Very easy to use. It's like, what day is it out there? When it is on the other.
Hello, welcome everyone. Hey.
Did you set up a document? Yeah.
Okay. Awesome. Welcome, everyone. Thank you all for joining us today. I just want to reorient everyone here. If you are planning to attend the point of use water quality and treatment session, this is it. You are in the right place. So I just want to start off by saying, we'll have our first presentation led by Camille Zimmer. Our second presentation will be followed up by Sarah Young and Julia Sluck. And our third presentation will be followed by Sabrina Hawk. Please save some of your questions towards the end of their presentations. Feel free to ask, take notes along the way, and enjoy.
Okay, thank you. Okay, I'm gonna use the mic because I don't like hiding behind the lectern. Can you guys hear me? Is that okay? Okay, good. Okay, so, thank you for the introduction. Just gonna start my timer. Okay, so my name is Camille Zimmer, and I'm from the University of Victoria in Canada. Yeah, I'm happy to be presenting to you my work on how well do point of use water treatment devices work. My supervisor is Keitano Doria, and I was also fortunate to work with a co-op student by the name of Alice Gentleman. So just to orient ourselves right now, so we're at UNC in yellow there on the left-hand side, and the University of Victoria in Canada is on the west coast of Canada on Vancouver Island, and as well Sombrio Beach, which is my fieldwork site, which I'll be talking about, is also on Vancouver Island. Just to get a zoom in, the red point there is Sombrio Beach. It's 2-hour drive away from University of Victoria. And they're down on the bottom left-hand corner, my left, your right. The bottom right-hand corner is Seattle and Vancouver up there. Okay, so I don't know if Marcio is in the room, but for those of you who are at the side session, Marcio pretty much gave my introduction for this. So thank you for that. So I apologize if I'm feeding you your own report that I screenshotted, but as we all are in this room knowing, 26% of the global population do not have access to a safely managed drinking water service. And so for those folks who do not have that, water takes a journey from the source to storage and consumption, leaving the potential for microbial contamination along the way, which can be mitigated through household water and treatment and storage. And although all aspects of household treatment and storage are important, my PhD work does focus on point of use water treatment methods, which is why we are here today in the session. So my PhD work aims to answer the question, how well do point of use water treatment methods work? And so as we talked about just before in this session, I may have asked a pretty self-serving question, but typically, as we were talking about, this question is answered in the lab. We saw this with the WHO scheme that was presented by Knox. It's evaluated in the lab by a science nerd, such as ourselves here in this room, by a method called challenge testing, where water is spiked with virus, bacteria, protozoans, or their surrogates, and treated in the lab under controlled conditions. And we can imagine that, and we can see somewhat in the literature that this is an overestimation generally of the performance of point of use water treatment methods. This is nothing new. Just to give a little thought experiment here, the bottom picture here of me, this is a backcountry backpacking trip I took in 2020. In this picture, I haven't showered for probably a week. My hands are not clean. I am using chlorine droplets to treat my water for drinking before consumption. And you can imagine that the protection I'm seeing from that water treatment, that those chlorine drops is nowhere near as good as you would be in the lab. So that feeds into my project objective, which is to take this lab challenge testing and bring it into the field. So how do we do this? The major hurdle is, of course, the challenge microorganisms, so the spike microbes. So to address this, What we did was in place of bacteria, or for bacterial surrogates, we used a probiotic supplement. So what's called, it's a brand name Mutaflor. It's available off the shelf in Canada, and it's safe for human consumption. It contains the strain E. coli missile as a surrogate for E. coli, and for surrogate for protozoans, we used baker's yeast as a surrogate for Cryptosporidium. And validating these microorganisms as surrogates is part of my PhD, but I won't talk about it here. So we're validating them via literature review and lab experiments. Okay, so what we actually did in the field, so I'll give you a quick overview. So I talked about where my field site was on the west coast of Vancouver Island. So we surveyed 144 backpackers who were camping in the Juan de Fuca National or Provincial Park on the west coast of Vancouver Island. The study context was heavily dictated by COVID-19 restrictions. My fieldwork took place in the summer of 2021, meaning that we were planning in 2020. The plan originally was to go and work with partners in Indonesia who were manufacturing ceramic water filters. That was obviously blown completely out of the water. So we did what we could. We looked locally to see who was using point of use methods in Canada, which happened to be backpackers in the backcountry who were camping and treating the freshwater sources for drinking. We did have an ethics approval for this. So each survey that we administered to backpackers consisted of a challenge test where we would spike a 1 liter sample of water from the stream at the campsite. We would spike it with that probiotic supplement I talked about, and also yeast. We would then hand that 1 liter of water over to the participant and ask them to treat that liter of water as they normally would, just whatever they would normally do with their own treatment device. They would not drink that water. We would then help them clean out their water bottle or flush their filter with clean water that we brought from home. So that's the challenge test. We did a hand rinse test to test fecal contamination on their hands. We also sampled their personal water and took a questionnaire. I won't present points 2 through 4. I'll only look at the results from the challenge testing here, but we do have that data. Okay, so here we have the meat and potatoes of my presentation. So we're looking at log reduction values, so orders of magnitude reduction of microbes. So a higher log reduction value is better. And what we found for E. coli, so for that probiotic supplement bacterial surrogate, is that nearly all brands that we studied performed worse than their manufacturer claim. So I'll start off by saying that manufacturer claim is a very broad term. So manufacturers, their claims, the data was anywhere from fully published with protocols and very robust to just a claim they had on their label. So manufacturer claim is a pretty broad term here, but we have what we have. So we looked at chemical disinfection methods. I won't get into the individual brands because we're in the US and I don't want to get sued. But yeah, so I'll start off with chemical disinfection. That's a fear that I had, honestly. So we'll start off with chemical disinfection. So things like chlorine tablets, for example. So one brand that we studied performed about the same as what they were claiming, and one performed significantly worse. We have 5 different brands of filters for which we have manufacturer claims to compare. And they all systematically performed worse than what their manufacturers were claiming. And then we have one method of UV disinfection that we studied in the field. So this was one of those like UV pens that you stick in the water and it shines the UV light. Yeah, so some things that are very striking here is that 6 out of 8 of these brands do perform worse than what they're claiming. And some of them have several orders of magnitude lower. And part of this is that almost all of them claim a 6 log reduction, which is actually quite high. So they're falling short of that. And so for yeast, so that protozoan surrogate, some of them actually performed better than the manufacturer claim, which is a little wild for us to see in our results. So for chemical disinfection, brand A actually performed better than what they had claimed, whereas brand B did not. All the filters performed, except for one, performed about what they claimed, actually. And for UV disinfection, it was right in the middle. Yeah, and so we're still scratching our heads a little bit, figuring out why this is, because in the literature, it does bear out that difference would hold, like that's poor performance in the field would hold for protozoans, and therefore their surrogates. A few ideas here is that we were testing, I mentioned that we had hand rinse samples to test for hand fecal contamination. As we can, those of us in the room who've been camping could probably use our imagination to see how dirty the participants' hands were. The contamination was quite high for E. coli in many cases. So this might be part of the reason why we're seeing that really low performance in bacterial reduction, but not so much with yeast, because most of us don't walk around with baker's yeast on our hands. And as well, the general claims for log reduction values aren't as high for Cryptosporidium as they were for bacteria. We have it was about 3 log across the board claimed for Cryptosporidium by manufacturers. So yeah, I'll finish off and open it up for questions after saying I just have one last case study, one last slide here. I did somewhat cherry pick this case study, not only because it really demonstrates the difference, but also because I had a really great picture of a dog that I took in the field. So this case study is for chlorine disinfection tablets, which are not Aquatabs, but a similar brand. So for bacteria, so our probiotic supplement, we saw in the field that it had about a 2.9 log reduction value, whereas the manufacturer was claiming a 6 log. For protozoans, we had an average of a 2.5 log reduction value, and the manufacturers were claiming a 6 log. We did not study viruses at all. We still are looking for a food safe viral surrogate. But yeah, we have this information based on bacteria and yeast. And if we were to make WHO scheme ratings based on these 2 bits of data, we would give it 1 star based on the field data and 3 stars based on the lab data. So it's a pretty big difference. So I'll sum up by saying field testing was successful, and it could be a useful tool to guide design and implementation. As Marcio said, what did he say? Supplement lab testing. So I would supplement Marcio's supplemental lab testing. Could be used as data inputs for health risk assessment. So instead of a lab log reduction value, we would be using field data. And as well used for quality control in decentralized manufacturing facilities. So we heard from Knox earlier that manufacturing variability can be quite high. So this could help monitor a little bit better that variability in manufacturing. Thank you. Yeah, question.
Hi, I was wondering if I know that's your different part of your dissertation, but can you comment on whether those are good surrogate organisms for Cryptosporidia? I mean, what have you found with your?
Yeah, so I'm actually my task that I'm supposed to finish by the end of next week is finish writing my my literature review on this. So I'm doing a literature review on disinfection and yeast. So we find that for UV, it's pretty good. Yeast versus Cryptosporidium and Giardia. For chlorine, there's actually not very much data out there. And of course, Cryptosporidium is a veritable fortress for chlorine. So it's not great, but it's also about the same as what other surrogates that we'd use. Yeah. And yeah, that answers your question.
Very, very interesting study.
Thank you.
Rhonda and Rotary. I spend most of my time in the field.
Yeah.
And it's often difficult to get these particular brands, but one thing we can always get is bleach.
Yeah,
And I wondered if you tested bleach and see how it compared.
that's a good question. So we didn't, basically, we were just testing whatever participants were using. And no one was using bleach to treat their water. Most of them were using chlorine, like the tablets. But that would be an interesting question for sure. Yeah, question.
You into like how old these filters were, or
Oh, yeah.
and also I'm thinking about use, like you go backpacking like twice a year.
Oh, yes.
It's gonna dry out as opposed to using it on a regular basis.
Yeah, so that was, we administered a questionnaire, which I'm not able to get into, but that was one of our questions on the questionnaire. And definitely the filter age was correlated with the log reduction value that we saw. We had people who were using filters that were over 10 years old. And replace your filters. Yeah, good question. Yeah.
Do you have any data on using these reduction methods back to the barrier approach, like filtration followed by chlorination?
Yeah, so for multiple barrier methods, we only had, so again, we were only looking at what people were using. So we only had, I think, 2 participants the whole summer who were using back-to-back methods. One was using 2 filters in a row, which was interesting. And then the other one was, I think she was a nurse and she was using chlorine and then filtration. And it worked pretty well, but it's kind of hard to say because it was only the 2 people the whole summer. Yeah. Question.
Thanks so much for your presentation. Obviously, you wanted your PhD on the beach. That's.
It was great. Yeah, I have so many pictures like this. Like, it makes it very easy to make presentations.
So in the development space, compliance is the big issue, right?
Yes.
And in terms of these differences in log reductions, can you speak a bit to the differences and how that relates to health benefit with regards to infectious doses infectious for Giardia or crypto? Like, is it really appreciably different when you're thinking about the risk of illness? A log reduction from 5 to 6, like, what does that really mean?
Yeah, like, does it matter? Basically, yeah. Yeah. Yeah, that's a really great question. So I think part of what we were asking participants in the survey was how easy or did they find it to use their device, and also how compliant, not how, we didn't ask them how compliant. I think we asked them like, how many times per trip have you drunk untreated water or something like that. So we looked at compliance, self-reported compliance, and self-reported how easy did they find the thing to use. So we did look at that. And we as well asked them if they had had any difficulties treating their water. So we did get a little bit of information on that to put together with the log reduction value. But yeah, it's true. Like how different is it really? And I think that feeds into, I'll go back. Oops. The second point, which is a health risk assessment. So I think that would be like, that's where this piece of data really feeds into a health risk assessment, is looking at that infectious dose. Yeah, thank you. Question.
Yeah, that it. Yeah, I think
We can
this,
chat at happy hour.
yes,
That's
if you
okay.
have additional questions, please find Camille after this presentation.
Yeah.
Another round of applause. Okay. And I'd next like to invite Sarah Young and Julia Flux for their presentation.
Awesome. Well, thank you so much, everybody. Standing between you and happy hour. So, but hopefully this will be interesting. My name is Julius Lux. I'm in the Department of Chemical and Biological Engineering and the Center for Synthetic Biology at Northwestern. And I'm joined by Sarah Young in the Department of Anthropology and the Global Health Program. We're going to tell you about a pretty exciting study that merges engineering and social science towards very much the vision that Camille was presenting about trying to really field validate technologies. In this case, point of use water quality detection technologies. Before we get started, I just want to acknowledge the two graduate students that worked in between our groups that really drove the majority of this and some of the funders of this work. And I should mention most of this work is the academic research that we've done, but I will at the end mention something exciting coming out of a startup company that I helped manage called STEMly. What we're going to do is tell you a little bit about the origin story, why we got interested in this project in the first place, and then tell you about the technology, which is really using this new field of what's called synthetic biology to develop point of use water quality monitoring technologies, and then tell you about the first real field trial of this that we just completed in Nakuru, Kenya, and then some exciting next steps. So I'll hand it over to Sarah.
Thanks. So I think we all have wondered that from one time or another, how do we know if our water is safe? Some of you may know that I work extensively on measuring people's experiences with water insecurity, and I'll just make a plug for our side event on Wednesday. Do come by if you'd like to talk about that. And in the course of developing these scales, I've seen a lot of water, and I've seen people wondering if water is safe or not to drink. And this water is obviously not safe to drink, but we all know it can look safe when it isn't. And as it happens, I have a great collaborator here because we've also collaborated on parenthood. And sometimes when our kids, including this one here, aren't interrupting us at dinner, we talk about science. And so I was pointing out how this is a concern. And it's a concern both for people making individual decisions about what water to drink or not, but as well as it's of interest to many of the agencies who are here today as we monitor progress or lack thereof towards the sustainable development goals.
So we got, Sarah was bringing home lots of photos about the challenges that people face in their daily lives, about where they get their water. And usually a common theme is you can go for the thing that's right there and it saves you time, but it's of questionable quality, or you can, put a lot more effort to go to a more clean source. And the concept was, can we empower individuals with information much more easier? I think familiar concept to everybody. Well, it's not like we can't measure water quality. These are some instruments that you might be familiar with, molecular biology or analytical chemistry to measure things from pathogens to chemical contaminants, metals, iron, things like that. The problem is these technologies just aren't scalable and affordable. Many of the instruments, as you know, are hundreds of thousands of dollars. They take PhDs to operate, or certainly a lot of training. And as a result, they work in a centralized infrastructure model. You have to have a lab, and basically that means you have to collect water and send it into the lab. And as we all saw with COVID, when we had to do that with the early COVID tests, it just doesn't scale very well, especially with water because it's heavy. This is starting to change a little bit. On the bottom, there's some online services, think of like Amazon for water quality monitoring, that are trying to scale this out, but they're still very expensive. And I should be fair, there's some other field deployable technologies that we'll mention. But tables like this really just motivated this concept of trying to make water quality monitoring accessible for anyone in the world. Our solution to this, or startup solution, is to go to this field of synthetic biology, which is generally the concept of, for our biggest challenges, can we ask, what does nature already do? And then ask, can we repurpose and re-engineer that solution? So it turns out for water quality monitoring, microbes have evolved lots of mechanisms to do this. The same things that are toxic to us are toxic to bacteria and yeast and everything like that. And the way that they deal with it, they've got these molecular machines called biosensors. These are molecules like proteins or RNA molecules that have evolved to bind to these contaminants. And in the cell, what they do, that binding event allows the cell usually to trigger some sort of gene expression process that allows the cell to mitigate it. Pump it out of the cell, chew it up, whatever you want. Now, we don't want to use engineered microbes for water quality monitoring. What we want to do instead is take these molecules out of the cell and repurpose them so that they work in a test tube and rewire them so that when the contaminants are present, we can produce a visual signal. Could be fluorescence, could be color, something like that to help us determine if our contaminant was present. I'm happy to go buy me a beer and I can tell you about all this works. We're going to talk about its effect. There are biosensors for all sorts of stuff right now. They're rapidly evolving. So these are some of the priority things. I've heard a lot of talks about this. You know, E. coli, fluoride, and arsenic are the top three we've heard a lot about, but metals, chemical compounds, you name it, biosensors are there. And we actually first got interested in this in the context of fluoride because we were studying how these things work in the lab. And we were reading articles, it's a little bit old now, but everybody here are the real experts, about how serious geogenic fluoride is. So long story short, we took one of the naturally occurring fluoride biosensor and we rewired it so that in this test tube, it makes a protein that makes a visible color. And this is just some of the data showing you various levels of fluoride concentration and these reactions after about an hour, you'll see a faint yellow color. And in this case, this is 2 parts per million, which is the US EPA limit, but you can tune these reactions to different sensitivities. And the rest, what I'm tell you, is tuned to the 1.5 parts per million WHO limit. As Camille said, this is great, works in the lab, doesn't work out in the field. This is our first foray into this. A while ago, we found a study in Costa Rica that was reporting high levels of fluoride near the volcano there. So this is one of our graduate students, Matt Verisloff, had the great pleasure of going to Costa Rica.
Seen in the beach.
Yeah, that's right, that's the theme here. And he just sampled a bunch of surface water, took some controls, all the controls work and found surface waters that had elevated fluoride. And here we're validating them using a fluoride electrode. Okay, and at this point, this is pretty exciting. Sarah and I started to write some papers, some primers on how you might do this for other contaminants. Scientific American picked it up because it's a really interesting, cool thing, some citizen science involved, and we're writing op-eds about, hey, everybody should be doing this. And actually right about here is where we were in 2019, the last time I came to this conference, with our kids in tow, doing Legos at the poster session, because, you know, science isn't enough for our kids. But the question is, does this, are they actually useful? Do you, you know, even though we took them out to the field, it was still a lab expert doing that. So I'll hand it over to Sarah.
And this is when the stars aligned for the two students who you saw at the beginning, so Walter and Patrick. So along the Rift Valley, geogenic fluoride exists, and it's terrible. I think I'll skip that background. And I'll say that the 2 questions that we sought to answer were these. Can real people use a fluoride biosensor test? We know a graduate student can on the beach. And then if they can use it, is it giving accurate information under field conditions? So the study design briefly is that we made the biosensors in Evanston, and that would be the royal we. I've never made a biosensor in my life. And we mailed those to Nairobi, got them to Nakuru, at which point we consented participants and conducted a survey with them about their knowledge, attitudes, and behaviors about fluoride and fluorosis, sociodemographics, and water insecurity. They then went and collected from 1 to 3 samples from their household, water samples, and tested those. You'll see more pictures of the test, but they put the water into these little vials. And then they were asked to look at the test every hour. They can take up to 6 hours, and in this particular iteration, to turn yellow. And then at that point, the field staff came back and did a second survey about the test experiences and interpretation, and did the gold standard test. So we were making sure that we were always leaving people with the correct information. This is what the test materials look like. So it was each sample takes 2 vials. So 1 vial for the water itself, and the second is a positive control. So we know that the tests are actually working. Yeah, so that's what it looks like being operated, and that's the fluorimeter in blue. So punchline is that real people can use these biosensor tests. So we are we had 36 participants who collected 57 water samples. They all could operate the test correctly. Two people struggled a little bit with their shaky hands, trying to drop one drop of water into the tubes. The tubes are a bit small. And then all but one test out of those 57 were correctly interpreted, which means that yellow was seen when yellow was observable and white was identified as correct.
Yeah, and in terms of the test functions themselves, this is a distribution of the measured fluoride concentration among the 57 samples. With the WHO line right there, you can see that in this particular case, a lot of the samples had high levels of fluoride. So over 79% were above the 1.5 parts per million, and the mean concentration was 6 parts per million. When we did the test though, and we had the validation data here, we could just make a little confusion matrix with the actual value being the fluorimeter measured value, and then the predicted would be, is the test yellow or not? And you can see that about a 93% sensitivity and a 75% specificity. So this was better than we expected, because this is still a pretty crude version of the format of the test. Summarize some of the findings. We're kind of adding another line to this table with fairly cheap tests that can be used at the point of use with no expertise. There's lots of other biosensors out there, as I mentioned. We did find some technology improvements, some simplification of the user interface is important, and also some interesting findings on temperature sensitivity. Turns out when you ship things to Kenya from Chicago, they go through Dubai, and there's some elevated temperatures there, and we noticed some degradation in the tests. We're working on that. The other thing is important about these. This is a scalable technology platform. It's a very simple thing. It can be mailed off in a little test kit. As a next step, this is where the STEM Loop stuff comes in. This technology is being commercialized, and STEM Loop 2 weeks ago launched an at-home lead and water test. There's an alpha tester program right now. So if you're interested in lead and water testing, you can go to STEM Loop and receive a batch of tests, as long as you provide some feedback data up there. The beta product, the alpha looks like it's 2 tubes, and it's this lateral flow strip. It works exactly like a COVID test. You put a drop of water in one tube, let it incubate, and then dip the strip in the other tube. These should be on the shelves pretty shortly with future contaminants coming out. Just to wrap up, this is us. Follow us if you like this type of stuff. These are some of the underlying papers behind this, and the results that we talked about are in revision, but there's a preprint available if you're interested. Thanks.
Hi, my name is Mook. I'm a grad student at LSE in London. So let's say a household in Kenya finds that there's fluoride in the water, it turns yellow. What happens next? Do they have any access to cleaner sources? Like what's next?
Yeah, that's a hugely important feature of all this stuff. You don't want to be delivering just problems, you want to be delivering solutions. We did provide filters to study participants. A more longer, but filters don't last forever. A longer term solution for these particular individuals was, where it is, we looked at the different types of water. The borehole water and the dug wells were the ones that had the fluoride, but the rainwater was basically fluoride free. So either rainwater sources or even dilution with rainwater, not ideal, but it is something that can be done.
I can add a little comment to that, having worked in Peru, facing this problem 10 years ago. And there we helped the homeowners build rainwater harvesting unit at the household level. Roughly, we provide them with the downspouts and the gutters and the masonry to build a 10,000 liter cistern. For $450, they then have a lifetime supply of safe water. We often add chlorine or bleach just to put the finishing touches to it, but so sustainable and minimal O&M expense.
That's great. There was another question here.
Oh, hey, my name is Katya. I'm at UC Berkeley. I had a quick question about for your tests where you're validating your biosensor against like an ion selective electrode. Did you test with a range of phosphate concentrations and other co-occurring ions? Because I've seen Other fields like lower cost methods for fluoride measurement really have a lot of interference and give inaccurate measurements, so there's a lot of phosphate in water specifically.
Great question. We did not look at phosphate levels. We did a bunch of tests in the lab to make sure the electrode could handle various water sources, Lake Michigan and things like that, but we did not do phosphate analysis. It's an interesting point.
I was working in Kibera, Kenya, and I had like a trans spectrophotometer or something, a handheld one, and I took my Mettler Guido and selected electrode. And it was never giving me the right measurement, and often it would say the water was contaminated when it wasn't, or above that 1.5 threshold. And measuring lower concentrations, it was, the ion selective electrode accepted, but the other ones were not.
Yeah, we found just the fact that you have to calibrate those instruments, and if you calibrate them wrong, they give you misleading results, which is a big challenge for those.
I'm just curious about how do you see these, let's say this becomes commercialized, how would you see it being used? What is the customer and how much would this cost? I mean, I assume it's targeted against people sort of at low levels of income, but.
Yeah, pricing is so heavily dependent on scale. The goal is to make it as accessible as possible. That's what I can say there. The consumer segment is kind of an interesting question. I think right now for the lead and water crisis that's happening and the billions of dollars US government is pouring into lead replacement, the target market is the big engineering firms that are trying to find where the lead pipes are in the country. So they would do tens of thousands of tests. And so that's the first target. If there is individual consumer interest and people want this test, which from what we can tell, is likely to only grow, then that will be another market segment to go after. And lead is the only the first product. This is an extendable technology to all sorts of other things.
So I would say it's useful for homeowners and individuals making their decisions about what lead lines to replace. But it's also useful for large scale monitoring. I mean, like imagine if mix can take along a couple of water samples with them and tell in 30 minutes what's in the water or not. So both of those. There were lots of hands that, yes, please.
I'm Emily, I've worked for Viridian, Spencer company. Did you ask or like do any surveys on people's interpretation of the data, if they understood it, or did they want to do that? Like that?
Yeah, we did. I mean, so obviously they were consented and we told them what we're gonna do. Then we asked what they knew about fluoride and if fluoride was a concern and if they were trying to do anything to mitigate it. And then we went on to do the testing and we talked about what the results meant, both in terms of what they saw and then, what the test reading was, as well as what the fluorimeter was saying. And it was clear that there was a lot of concern about fluoride, fluorosis. And there was a lot of misperception about how to treat it. So there's a lot of boiling that was happening in order to get rid of fluoride, which obviously, and at a third point, but yeah. So yes, this was a concern and this was information that was useful.
I'm Esther from Unison, and we're working on rapid response testing, as I'm sure you guys know about. Sorry, my question to you is, in the Kenya testing, how did you see the 6-hour time result? So you've got that little bit of kit, and your consumables are what, less than $1, whatever it is. But that piece of hardware had to sit there for 6 hours in the household, and then what someone came to do. How do you see the hardware piece fitting into this testing methodology?
Thank you for the question. And just to clarify, the hardware is not part of the at-home test kit. It's just the photometer. So the study coordinators had one photometer and they kind of went around and used it. But the test kit is just the little tubes that were on the left side of the slide. Yeah. This is what, I mean, these three are just packaging bags. These are 3 tests, and then these are little 3 20 microliter pipettes. So that's all that sits there, and they just sit, in ambient conditions.
The biosensor is the hardware.
Okay, right, that makes sense. And then how much water sample is it?
It's 20 microliters. So you only need a little drop, which is actually a challenge. And so future iterations is going to try to bump that up, because getting a 20 microliter drop in the tube is one of the pain points. Yeah.
All right, can we
Thank you.
please give?
Stickers up here about the lead taps, you can find out more.
Okay, so if you have any other questions, please feel free to find them later or at happy hour over a beer or wine. Or if we don't say, we'll tell you time. All right, and now we have Sabrina Haas. Floor is yours.
Hi, so good afternoon. My name is Sabrina Hawk. I'm presenting results today from a 30-month assessment of a filter intervention that was implemented through our Ministry of Health program in Ramayana, Rwanda. And so I worked on this research while I was a doctoral student at Emory University, along with co-authors that were on my PhD dissertation committee and collaborators in Rwanda. One of those co-authors, Matt Freeman, in the back of the room. Say hi, Matt. He might ask me where the manuscript is for this study. Anyways. So for today's outline, I'll start off with a brief introduction and motivation in the study context. I'll then discuss the main components of the intervention design. And then I'll give an overview of the study methods and then go into the results and discussion. So at a very high level, our motivation for this study was to really address the implementation gap of delivering household water treatment and safe storage interventions. We have a number of rigorous studies that show that they are highly effective in improving microbial drinking water quality and reducing diarrheal disease burden. But often there's limited examples of scaled use in populations that could benefit from them the most. So in sub-Saharan Africa, analysis of DHS data showed that about just 22% of households actually reported clean their drinking water. There are several reasons for barriers to scale, from just the intensive behavior change that's needed, access to efficacious treatments, political will, but today I'll be mostly focusing on sustainability. So although we do have a lot of trials showing that they are effective at improving microbial drinking water quality and reducing diarrheal disease burden, they're usually short in duration. So these interventions are seldom evaluated past 1 year. So studies that are over 6 months in particular usually show decline in effectiveness stemming from a decline in just use and coverage. And so this raises questions on just the sustainability of scaling HWTS interventions and whether this is a worthwhile investment. So our study on sustainability is a part of a larger evaluation. We conducted this within Rwanda's community-based environmental health promotion program. We first did a randomized control trial among about 1,200 households. We delivered LifeStraw Family 2.0 filters to a random half across 60 villages and followed them for about 15 months after the delivery of the intervention, both control and intervention households. We found that this intervention reduced the prevalence of fecal contaminated drinking water by about 20%, and the prevalence of moderate and higher fecal contamination by about 35%. 7-day recall of diarrhea was also reduced about 50% among children under 5. So after the trial, we followed the intervention households for an additional 15 months. This is about 608 households. So this is over a total of 30 months of follow-up in total. We again measured uptake, water quality as measured by E. coli presence, and child diarrhea prevalence. And so again, we did this to examine the filter's real-world sustainability. The filter itself is engineered to be microbiologically effective for at least 3 years, but there are limited assessments of their programmatic sustainability over 1 year. And so we are presenting the results of this today. So on to just a little bit about the intervention and the technology. The technology itself was, again, the LifeStraw Family 2.0 filter. This is a tabletop filter that uses ultrafiltration to filter out viruses, bacteria, and protozoa. Treated water dispenses through the safe water storage tank that also has a tap, and that prevents further contamination. Households were mostly instructed to actually keep water into the safe storage tap and to drink directly from it. But oftentimes we would see households actually storing treated water in another type of vessel. So again, the filter was delivered in the context of Rwanda's community-based environmental health promotion program. This is a nationally scaled program that uses community hygiene clubs to promote improved wash behaviors. These clubs are administered at a village level. They can have up to 100 members, and they meet about 1 to 2 times a month to discuss some of the curriculum of different wash behaviors. So filter training and messaging was integrated within the existing CBHP curriculum. Community health club facilitators and environmental health officers were trained to be basically the service provider of the filter, teaching the households how to use the filter and support the household if any troubleshooting was needed. There was a mass filter distribution event among the 30 selected villages to eligible household members who were members of the community health club and also had at least a pregnant person in the household or a young child living in the house. These filters were given free of cost, so fully subsidized. Next, the community health club facilitators did 2 follow-up visits with households in the next year. The first one was immediately after the mass distribution event, basically a one-on-one to teach households how to properly use the filter. And then a second one was done about 6 months after, just as a maintenance visit to see if anything needed to be repaired and just another reinforcement of how to use the filter. There's also ongoing promotion during the community health club meetings that I mentioned. And then finally, there was a repair and replacement process implemented just for non-functional filters in the first year. It was just a very small number of households that actually needed filter replacement. The implementers of this intervention were Catholic Relief Services and SNV Rwanda. They are 2 of the government's partners that implement community-based environmental promotion on the government's behalf. And they were also helped by Maziego, which is a social enterprise that had a lot of experience in delivering these filters in Rwanda. Briefly on our methods. Again, the research question we were asking, does filter coverage, condition, use, and the prevalence of contaminated drinking water and child diarrhea vary over the course of 30 months since the delivery of the intervention? So our data collection, it started in 2019. Again, this was first in the context of a randomized control trial that enrolled 1,200 households. 608 of those households were in our intervention group. We did our delivery of the intervention in March 2019 to June 2019. About 6 months after, we had our first follow-up, and about 9 months later, we had our last end-line follow-up. This was actually delayed because of COVID lockdowns, and so it ended up being 9 months after delivery, after the last follow-up. After this follow-up that concluded our trial, we actually lost our control group and we delivered the filters to the control households. But again, we still did continued monitoring over 2 more household visits for the next year. The third occurring in March 2021 to June 2021, and the last from October to January 2022. These follow-ups had a different, coincided with different rainy and dry seasons. And that was important for us just to understand how that might affect microbial water quality. So on outcomes, we measured coverage, whether the filter was actually observed to be in the household, the condition, whether the filter was assembled properly, didn't have any breakages, leakages, had a working tap, had a good flow rate. And then we also looked at reported use and observed use. Reported use, just simply asking households if they were using this life straw as a water treatment method. And then observed use. We actually looked to observe if there's actually water present in the storage container of the filter. And then finally, we looked at water quality and child diarrhea. For water quality, we measured point of use water samples, so meaning that we asked the household to give us a glass of water, and that is what we tested. We looked at E. coli indicators, and then we also looked at child diarrhea, looking at reported 7-day recall of WHO-defined diarrhea in children under 5. So our statistical analysis. We estimated prevalence ratios on binary indicators on uptake, water quality, and diarrhea, where we compared outcomes from the first follow-up visit, which is that 6-month visit, to later follow-up rounds.
using Laplace generalized estimating equations, and adjusting for village level clustering, to see these prevalence ratios. And then we further adjusted for seasonal factors such as average monthly rainfall, average monthly temperature, household size, water treatment at baseline, and socioeconomic status. And then diarrhea models were additionally adjusted for age and gender. So from our participant flow, we detected very minimal loss to follow-up. We had over 92% of possible household observations analyzed, so a little bit of attrition throughout the study follow-up period. We did have missing water samples just due to water scarcity, where we only could do 75% of possible intervention water samples. Often the time, households just simply did not have water to provide in their household for us to actually test. So presenting the overall results on coverage, condition, and use at 30 months post-delivery of the intervention. So by the end of the follow-up, over 94% of households still had the filter. 85% had a filter that was observed to be in good working condition. 85% reported to use the filter. And then 59% of households reported were observed to actually have water in their storage container. And so I'm not presenting the individual models that we have on each of these uptake outcomes, but basically rates on filter condition and use were comparable between the 6-month and the 12-month visit, but did have a significant decline when you compared it to the 24 and 30-month visits. So we did see a decline in this uptake in the second year, particularly. But looking at fecal contamination and child diarrhea, the prevalence of clean drinking water, that is water samples that did not have any indicators of fecal bacteria, compared to the 6-month visit, which is right here, where 27% of samples actually were not contaminated with fecal bacteria. Water quality did not deteriorate compared to the 6-month visit. Water quality did seem to get a little better at the 15-month visit, which is what this is showing right here. But overall, we did not see any decline in water quality compared to that first 6 months. Very similar to child diarrhea. We did not see any increase in the prevalence of child diarrhea since the 6-month visit. It did decline at 24 months, but overall, it did not increase over the 30 months. So discussion, I think one of the first questions that I had, so if use declined, why didn't water quality decline? So it's difficult to say without analysis of who stopped using the filter. Use does not necessarily mean correct use. So there was a still large proportion of households that did have contaminated drinking water despite reported or observed use of the filter. And so it's seasonality related. I'm not presenting this data, but the control group also had continued monitoring after they received the filter at the conclusion of the trial. And so they actually did have similar water quality and diarrhea outcomes in their first year of exposure compared to the interventions group's second year of exposure. So this allows for some contemporaneous comparison. So in conclusions, so the coverage of the filters were high throughout our follow-up period. Indicators on condition and use seem to most have declined in the second year of follow-up. This could reflect the importance of that maintenance visit in the first year. And the effects of water quality and diarrhea seem to sustain over the course of the 30 months since that 6-month follow-up and intervention delivery. And results seem to be more favorable than some other filter studies that did have comparable follow-ups. So this might mean that community health clubs, because they are a sustained village institution, that could perhaps had an important role in promoting and sustaining the technology. Just some limitations, the generalizability of the results, it's exclusive to Ramagana districts and members of community health clubs. We're also only able to make conclusions on this particular filter technology. A lot of sustainability questions still exist on other HWTS interventions. And then also, this was also a fully subsidized intervention. There's also potential for reporting and courtesy bias, and it relies on self-report filter use and health outcomes. that we did have the missing data on water quality samples, which also could be a source of bias. And we relied on an observational study design without a true counterfactual. We attempted to adjust for seasonality factors and utilized the trial design when possible, but still. And so then the last 3 follow-ups also did coincide with the pandemic. This seemed to actually interrupt some of the CHC club meetings just due to lockdowns in the country. But some strengths, this is one of the longest follow-up periods for this filter, and it provides improved insight on sustainability. This is longitudinal data collection over multiple dry and rainy seasons. And then this is a piece of implementation research where it was evaluated under real world conditions in the context of an existing public program. And so the research team did not design or deliver the intervention. Community hygiene clubs are a very popular service model in at least 17 other countries, so they could be replicated. With that, thank our collaborators in Rwanda, from Emory University, and for our funders. And with that, I will take questions.
How much water did you need to collect and how did you test for E. coli? That's really a piece of information if there's no water in the house.
Yes, so we would take 100 milliliters of sample water, just according to how the mix would measure water quality. And yeah, that was always something that was striking of, there was at least 25% of households that just didn't have water in their household to actually provide us. And not just from the filter, but anywhere in their household. And so that's a limitation, of course, and that's something we didn't expect.
Also information.
Yeah, and so we do at least have water samples from every household, at least for one of the follow-up visits, but it is a limitation. And we also did collect a lot of HOI's data along with it, because that kind of inspired that as the missing water samples. Yes.
Thank you for your presentation. So jumping on this question, were you able to identify if the water sample was from the water filter as well? Like, were you able to identify like the percentage maybe of houses where they did use, like, you know, for sure that the sample was taken from the filter as well?
Yes, we do have that data. I'm not presenting that today, just because we were wanting to look at our primary outcome of just point of use water, but we do have what we have, where that water was actually stored, if that water was actually coming from the filter, treated with the filter, and then importantly, if they were actually taking water from the filter directly into their glass of
Did
water.
you see a difference or?
Yeah,
Like, yeah.
yeah. Yes, that is one of the drivers of contamination of just how they actually store the water. Yes.
Did you say at the beginning that there was a reduction of incidence of diarrhea found?
Yes,
And what was that percent?
so for the 12-month randomized control trial that we did, it was a 50% reduction
50%.
in diarrheal presence with that ones. So between the intervention and control groups.
Thank you.
I think you had your hand up first.
The E. coli data you have, it's the cutoff of 1 CFU per 100 mil, which is a pretty high bar. So I'm curious, do you have quantitative data above that? Like, if you were to look at a different level, do you see any difference in the result?
Yeah, we did ensure to have binary indicators on all 3 levels of WHO risk categories. So we have it on just all fecal contamination indicators of fecal contamination where they had to have less than 1 CFU 100 milliliter. But we also looked at moderate and higher, which is less than 10 CFU per 100 milliliter or less than 100 CFU. And that I think that of course did have a much more of an effect on those risk categories. But it didn't have this data in there today.
Sorry, meaning that it didn't have the same, can you just say more about that? It had an effect on the risk category.
Yes, so we saw from the trial about a 35% reduction in moderate and higher fecal contamination, and then a 45% reduction in very high levels of fecal contamination.
But then
Of this portion of them. Over time, I don't have the data with me right now to disaggregate it by, but yeah, I can connect with you offline. You had your hand up.
Congratulations on your 30 months follow-up. What that frequency of these from this line area, so it also is kind of much smarter. That household, that big household that the 12 months. If we see kind of that. And the second thing, the almost 15 minutes of aluminum and how we use that on.
Yeah, so on frequency of use, that was a bit trickier to really quantify. We did ask some quantitative measures on basically how many times did you actually fill this filter in the past 7 days? So we do have data on that. And the frequency did seem to decline over the course of the months. Sorry, and your second question on looking at why they might have stopped. We did some responses on why exactly did you stop using the filter. Some people either just didn't have a filter in their household and they sold it, or it broke. And then some people just, I guess, lost interest in using it. But yeah.
Can we please give a round of applause to all for so this concludes the end of the presentations. Feel free to head to the atrium to see the posters and the happy hour reception. And of course, if you have any questions to the speakers, come up. Sorry, you might get inundated, but I think it's a good thing, right? All right.